So let's look at peptides that actually get loaded onto this allotype. And so you know already where peptides are loaded, how peptides are formed and loaded onto MHC class 1. Hopefully you know. Peptide binding motif, that's the other term that we talk about when we talk about which peptides can be presented. I forgot to mention that. So the peptide binding motif for allotype A0201 is a leucine at anchor position 2 and a valine at anchor position 9. So let's go to the endoplasmic reticulum. Right? That is where peptides are loaded onto MHC class 1 molecules. Right? And so we know how peptides get into the ER. Right? The proteins in the cytoplasm are broken down by the proteasome into peptides. The TAP transporter pumps those peptides in. Right? And peptides get loaded. Now, I just said peptides get loaded before, but what I really meant is peptides get loaded on the isoforms of the MHC class 1s that can bind those specific peptides. So in this instance, will every peptide get loaded onto A0201? Absolutely not. Only those peptides that have the that match the peptide binding motif will get loaded onto this isoform. So if out of all those peptides that are in the ER, some of them have leucine at position 2, valine at position 9. Those peptides will get loaded onto this isoform. The more peptides that can get loaded, the better the antigen presentation. Because, for example, if those red peptides represent viral proteins, then the more viral proteins we can present, the better our chances that a T cell will recognize this as an infected cell. So it's all about presenting more peptides, as many as possible. So that brings us back to, let's talk about the fact that, what if you couldn't present any of the peptides? Let's say those blue ones are self-proteins and are self-peptides, and the red ones are viral peptides. What if none of them match? What if none of the viral proteins had a leucine at position 9 and a valine at position 9? Leucine at position 2, valine at position 9. What if you couldn't present any of them? If that's the only allotype you have, if that's the only isoform you make of the MHC1 molecules, then you couldn't present, and you couldn't tell the immune system that the cell is infected. So what's the solution? The solution is to have more versions of MHCs, right? And that brings us back to the fact that there are many different versions or allotypes of the HLA-A gene. So we just talked about allotype 0201, and it can load a specific peptide with a specific peptide binding motif. Now let's talk about another real allele or allotype of HLA-A called A3001. Right? The peptide binding motif of that allele, 3001, is different. It has phenylalanine at position 2 and phenylalanine at position 9. So any peptide that looks like that can get loaded into 3001 isoform. So let's go and look at that a little more closely. Right? So here's now the allotype A3001. Right? So it's still an MHC class 1 alpha chain, right? But the amino acids encoded for it in this allele or this allotype will allow only peptides that have the phenylalanine at positions 2 and position 9 to be loaded into it, right? So this peptide here, this sort of random peptide I've drawn, it's got a phenylalanine at position 2 and 9. So it can get loaded into this peptide binding groove, right? And there's that interaction between the anchor residues of the peptide and the amino acids present in this allotype, this isoform, 3001. So let's go back to the endoplasmic reticulum. Now, instead of showing one allotype or one isoform of the protein, we're going to show both. Right? So again, let's say we're infected by a virus. Here's a viral protein made in the cytoplasm, right? And the virus is just trying to replicate. So the cell's going to steal some of that protein. Right? This protein has some specific amino acid sequence. The cell tries to steal a little bit of that protein, sends it to the proteasome, chops it up, right? And sends the peptides where? Into the ER. And hopefully we can present some of these peptides to a CD8 cell to show that we are in fact infected. So which peptides can we present? Well, we can only present peptides that bind are isotypes or isoforms. So we know that 0201 will bind any peptide with a leucine and a valine at those anchor positions. Well, now we have another one. We have A3001, which will bind any peptides that have phenylalanines at both those anchor positions. So, for example, let us say that in this real viral protein, there are some phenylalanines, right, in certain positions and some leucines and valines at certain positions. Well, that's great. We can present those peptides. If there's a peptide that doesn't have those amino acids at those positions, let's say here's another peptide that has arginines at position 2 and 9. That peptide not going to get loaded onto those isotypes. But the other two peptides, those will get loaded onto those isoforms of the HLAs. So that's good. We can present those. So out of the 6 peptides we generated, we can present 2 out of the 6. And hopefully we will find a T cell that recognizes one or both of those. And that would be great. So, but clearly it would make a lot more sense if we can present as more peptides. If we can present all of those peptides, then our chances of being identified by a T cell receptor would go up greatly. So the more peptides we can present, the better. Well, how are we going to present more peptides if these are the only 2 MHC class 1 alpha chains we can make? The answer is we can make more. Let's go to the next slide. So I just told you before that we have the HLA-A gene that makes the alpha chain protein, and you most likely have different allotypes. Well, what I'm now going to tell you is that there was gene duplication somewhere in the course of human evolution, and we in fact have multiple versions of these genes. So HLA-A, HLA-B, and HLA-C. All of those genes encode for the alpha chain of a MHC class 1 molecule. So in fact, you could have up to 6 different versions of the MHC class 1 alpha chain because they are encoded for by these 3 different genes. You have 2 copies, one from mom, one from dad, and so you can make up to 6 different major HLA alpha chains or MHC alpha chain proteins. So we call this, there's a gene family. For most genes, there's one version, there's one gene, and you have 2 alleles, and they make the proteins. So you can have 2 different versions of that protein. For the MHC alpha chain, you in fact have 3 different genes, A, B, and C, the major ones that make the alpha chain protein. And since you have 1 copy from mom and 1 copy from dad, you in fact can make 6 different versions of the alpha chain. Which basically, what's the advantage of this? You can present more peptides. And the more peptides you can present, the more likely you are to, in fact, survive a viral infection. So the next video is going to give an example of an MHC isoform encoded from the HLA-B gene. And so I'm going to talk about another gene, HLA-B, that's going to encode for the alpha chain protein. And we're going to see how that allele is going to help us present more peptides, right? So when you think about the fact that your cells are covered in MHC class 1 and they're presenting peptides, but in fact they are covered in different MHC class 1s. And so this is bringing us to this concept of diversity. The more diversity we have in our immune system, the better we are to present viral peptides, the more likely we are to survive a viral infection. So in the next video we'll talk about the HLA-B gene.